Hello everyone, welcome to Groups Master. This is APPSC Group 1 Mains Revision Series Science and Technology. ఈ వీడియోలో మనం టాపిక్ 8.5 టిష్యూ కల్చర్ మెథడ్స్ అండ్ అప్లికేషన్స్ డిస్కస్ చేస్తాం. దిస్ ఈజ్ అవర్ అఫీషియల్ వెబ్సైట్ groupsmaster.com. ప్లీజ్ విజిట్ దిస్ వెబ్సైట్ ఫర్ ఫర్దర్ అప్డేట్స్ అండ్ ఇన్ఫర్మేషన్. టిష్యూ కల్చర్ మెథడ్స్ అండ్ అప్లికేషన్స్లో వాట్ ఈస్ టిష్యూ కల్చర్ అని ఏదైనా క్వశ్చన్ వస్తే కనుక టిష్యూ కల్చర్ ఈ విధంగా ఇంట్రడ్యూస్ చేయొచ్చు. టిష్యూ కల్చర్ ఇస్ ద ప్రాసెస్ ఆఫ్ గ్రోయింగ్ అండ్ మెయింటైనింగ్ plant or animal cells outside their natural environment under controlled laboratory conditions. There are several tissue culture methods used depending on the specific purpose and type of cells being cultured. Here are some of the common tissue culture methods. First one is callus culture. In this method, a small piece of tissue, usually from a stem or root, is taken from the plant and placed onto a nutrient medium containing hormones and nutrients. Then the tissue develops into a mass of undifferentiated cells. దీన్ని మనం callus అంటాం. This technique is commonly used for plant breeding and genetic engineering. Next one is suspension culture. In this method, cells are grown in a liquid medium that is constantly agitated. This creates a suspension of individual cells that can be used for the production of vaccines and therapeutic proteins. Next one is explant culture. This method involves taking a small piece of tissue such as a leaf or stem and placing it on a nutrient medium. The explant grows into a new plant or tissue that can be used for research purposes. Next one is organ culture. In this method, a whole organ such as heart or liver is placed onto a nutrient medium that provides the necessary nutrients and growth factors. This technique is used for studying organ development and function. Next one is embryo culture. This method involves the cultivation of embryos into a nutrient medium. It is used in animal breeding and for studying embryonic development. Next one is hairy root culture. In this method, a bacterium called Agrobacterium rhizogenus is used to infect plant tissue, causing the formation of hairy roots that can be used for genetic engineering and the production of secondary metabolites. దీన్ని మనం ఈ విధంగా కన్క్లూడ్ చేయొచ్చు. These tissue culture methods are important for research in fields such as genetics, biotechnology and medicine. They allow scientists to study the behavior of cells in a controlled environment and to develop new techniques for the production of useful compounds and materials. Next, applications of tissue culture. The tissue culture methods are important for research in fields such as genetics, biotechnology and medicine. They allow scientists to study the behavior of cells in a controlled environment and to develop new techniques for the production of useful compounds and materials. One of the application of tissue culture method is plant propagation. The tissue culture is commonly used for the mass production of plants. It allows for the rapid production of disease-free, genetically identical plants that can be used in agriculture, forestry and horticulture. Next application is drug development and toxicity testing. Tissue culture is used to test potential drugs and assess their toxicity. Cells can be grown in culture and exposed to various drugs or toxins to determine their effects on cell growth and function. Next is tissue engineering. Tissue culture is a key technique in tissue engineering which involves the development of functional tissues and organs for transplantation or research. Tissue engineers use tissue culture methods to grow cells and tissues in vitro and then engineer them into functional tissues and organs. Next one is regenerative medicine. Tissue culture is used to develop new treatments for diseases and injuries by growing and transplanting healthy tissues and organs. For example, stem cells can be grown in culture and then transplanted to replace the damaged or diseased tissues in the human body. Next application is genetic engineering. Tissue culture allows scientists to manipulate the genes of plants and animals, creating new varieties or modifying existing ones. This is used to improve crop yields, develop new medicines and create new varieties of animals for research. Next application is production of secondary metabolites. Tissue culture can be used to produce secondary metabolites which are important for the production of medicines, flavors and fragrances. This allows for the sustainable production of these compounds without the need to harvest plants from the wild. Next application is disease diagnosis. Tissue culture can be used to diagnose diseases by isolating and culturing pathogens from the infected tissues. This allows for the rapid identification of causative agent and the development of appropriate treatments. Next application is cancer research. Tissue culture methods are used to study the growth and behavior of cancer cells. This allows researchers to develop new treatments for cancer and to better understand the underlying mechanisms of the disease. These are some of the applications of tissue culture. Thank you very much. If you really like this video, please subscribe to Groups Master YouTube channel. Thank you.